The small intestine. So the small intestine is going to be our main location of both chemical digestion and absorption. So 3 segments. You have the very short initial segment, maybe 15 centimeters long, the duodenum. Then you have the first roughly 2/5, which would be the jejunum. So between the duodenum and the jejunum, that's where we see the majority of both chemical digestion and absorption. And in the ileum, the longest portion, we see most of the reabsorption of the water that we've pushed into the small intestines. The duodenum is the location where we see the bile duct and the pancreatic duct empty into the small intestines. And there's a small hepatopancreatic sphincter that controls the main entrance from those 2 areas into the duodenum. So if we look, we can see this little C-shaped segment of small intestine would be the duodenum. From the liver, you have the 2 hepatic ducts, which combine to form the common hepatic duct. From the gallbladder, you have the cystic duct. When all of those combine, now we're in the bile duct. From the pancreas, you can see the pancreatic duct, main pancreatic duct. So the bile duct and the pancreatic duct come together at the hepatopancreatic ampulla, and there's a sphincter that controls release into the duodenum at that point. Worth noticing, there is an accessory pancreatic duct, so pancreatic secretions can come through that, and this is an unsphinctered opening into the duodenum. Internally, in the mucosa, we see a lot of structural modifications that increase surface area. And that's going to be important for nutrient absorption. So we have folds on folds on folds. So first off, you have the plicae, which are the macroscopic, the gross circles that you can see inside that mucosal space. On top of that, we can see the individual villi. And then if we were to look microscopically on top of those villi, there's microvilli, which create another level of folding. So lots of surface area. On top of the microvilli, in addition to being the site where absorption takes place, there's also a lot of brush border enzymes where the finished portion of chemical digestion occurs for most nutrients. So those microvilli, that's where we see those brush border enzymes. We also see the retention of, so we don't call them pits in the small intestine, we call them crypts, intestinal crypts. And we start to see, these are where the secretory cells are that produce intestinal juice. Intestinal juice is really just water. We see endocrine cells, and then we see these types of cells that produce, so you have the IELs, which try to destroy infected cells, and the Paneth cells, which secrete antimicrobial agents. And so we're creating this barrier between what's in the lumen and what is inside our body. So this shows that lining. So this would be the villi. And then as you go into this crypt, this is where we would find in green the enteroendocrine cells. In this crypt, we also see an increase in, so this is called Mucosa-associated lymphoid tissue or MALT tissue. And we see lymphatic vessels that reach up into these villi. And that's all part of the lymphatic system, which is going to help us fight off any potential infections.